can they do anything in an animal? Do they really do anything that can matter for us? So there are a couple animal models that really are the gold standards in this area. One is the RCS rat, the Royal College of Surgeon rat, and they develop macular degeneration that's very similar to humans. And so what we went in and is we used some of our human RPE cells that we made, and we tested them in these animals, and then we could measure the visual acuity of these animals. And so if you look at this, you can actually see here, this is the visual acuity. As you increase the number of cells to a higher and higher dose, you can see that there's a dose escalation, and that by the time you get to these higher doses, you see 100% improvement in visual acuity over the untreated animals. Similarly, there's a mouse that's similar to the Stargardt's disease, and we can show that the injection of these cells prevents the onset of the disease. But what I find more impressive, and certainly as a scientist, is that when you look at the histology of these animals, that is that when you look at the eyes after the animals have died or are terminated, you can actually do a cross-section and look at what's going on in the eye. So in the untreated animals, what you're seeing here, hard to see, is one layer of photoreceptors left. These animals are blind. They're essentially blind. They can't see. However, in the animals that got the injection of these RPE cells, this is that layer. This is the outer nucleated layer, 5 to 7 layers thick. So we had very significant rescue of these photoreceptors. And this is exactly the process that goes on in people who have macular degeneration. So all this data was put together, presented to the FDA. Now the FDA, in your first clinical studies, isn't so much concerned about what you can do to help the patients as it is, well, first to show that your technology is safe, that you're not going to do any damage. And it turns out that embryonic stem cells, by definition, when you inject them into the body, they have to form tumors. They have to form these things known as teratomas, and they have to turn into all sorts of things. You can actually look at these teratomas, and they will turn in, and they will have hair and bone, and some of them even have whole jaws. So they turn into everything. And of course, when you put something into the eye and you want it to be a retinal cell, you don't want a tooth. So it was very important to the FDA that we don't have these undifferentiated cells. So what we had to do was develop a method to show them that wasn't the case. So we developed an assay where we could detect even one of these undifferentiated cells in over 1 million cells. So if we had 8 million cells, we could throw in 5 and find all of them. And it turns out that actually for our therapy, the highest dose was 200,000 cells. So we could actually discern even if we had 1 cell. But more importantly, even if you throw in 10% of these undifferentiated cells at the end of the manufacturing process, you still don't end up with any. So that was good. We could say that, we can look at these cells in the dish and know that they should be safe. But the FDA says, well, that's not enough. You got to go into animals for their lifetime, and you need to show us that your calculations and everything are right, and that you don't get these teratomas, because that's what their main concern was. And indeed, we went in and we did that in these animals, and not only that, but they wanted us to actually spike in large amounts of these undifferentiated cells to see, even if we were way off, like 6 orders of magnitude, will we get a teratoma. And indeed, we didn't see any tumors in any of our animals. We studied hundreds of these animals. Not only did we not see any tumors, but we didn't see any ectopic tissue. That means we didn't see any other cell type other than the cell type we put in there. So based on that, the FDA gave us approval to do the first studies to treat macular degeneration using this technology. We have 2 ongoing trials, again, one for dry AMD and the other one for Stargardt's. We have 4 clinical sites. One is Mass Eye and Ear in Massachusetts. The other is Bascom Palmer in Florida, Wills Eye Institute in Philadelphia, and Jules Stein at UCLA out in LA. So we've studied, to date, we've treated 12 patients with dry AMD. They have various amounts of the cells. We've treated 9 patients with Stargardt's disease, and we've treated 9 patients in our European trial, which is being carried out at Mooresfield Eye Hospital. And this just shows you the design of the clinical trial. Each of these clinical trials is 12 patients, so 12 Stargardt's, 12 AMD in the US, and then also 12 Stargardt's in Europe. So we've treated 30 patients to date, and we also recently got approval based on the safety to actually move into patients that have better vision. And the goal of this technology isn't really to reverse blindness. It's that what happens is that when the patients start to lose their retinal cells, when they lose their retinal cells, you no longer are maintaining the healthier photoreceptors. So our goal is to get into patients before they become blind, before they lose the vision, and to put these new healthy RPE in to prevent that from occurring altogether. So we've just now started to go back into these younger patients or patients that have better vision to see whether we can halt that process. But the first patients were mainly to see whether or not it was safe, and these were advanced patients who already were legally blind. And so the trial design was that you start with a low dose of cells. We started with 50,000 cells. We treated one patient, and then after we showed it was safe, we wait 6 weeks. It goes to an independent data safety monitoring board, and if they assess and say everything looks good, they let us do the next 2 patients. And then we go through the same thing. We double the number of cells to 100, and we've now finished more or less our 150,000 cells. So we published the results of the first 2 patients early last year in the Lancet, and we showed in those first 2 patients that first of all, we didn't see any safety signals, but in the first Stargardt's patient, for instance, she could only detect hand motion. Within 1 week after the injection of these cells, she could start counting fingers. Within 2 weeks, she could start counting letters on the visual acuity chart. In 1 month, she could count 5 letters on the visual acuity chart. It's now been over 2 years, and she's getting better and better, when in fact, she should be getting worse and worse. And so that was very encouraging. And the good thing about the eye is that you can actually look into the eye with instruments in real time and actually see what's going on. So you don't have to wait till the patient dies to know if something bad is happening or what's going on with your cells. And it turns out there are instruments called OCT, for instance, that are very high resolution, and you can actually see individual cells. So here what you see are our RPE cells attaching to Bruch's membrane. That's what they're supposed to attach to. So those were the first 2 patients. And so, as I mentioned, we've now treated 30 patients. So overall, we're in the process of putting together the publication, so we're not allowed to really tell you too much of the details. But what I can say is that we haven't seen any safety issues whatsoever related to the stem cell treatment. This is really important because we're the first ones doing this, the only ones in the world, so if we mess up, it would ruin the whole field. So that was a good thing, that nothing went wrong that we could see. Secondly, is that we're seeing very clear signs that the cells have engrafted and survived. So what you're seeing here, this little dotted area, that's the injection site. That's where the bleb of cells is that we put. And what you see is after 2 months, you see these little pigmented patches here appearing, and then they get even more pigmented with time, in this case 6 months. So those are cells that are clearly living and engrafting and staying alive. And during the 1-year follow-up period, the patients, both the Stargardt's patients and the dry AMD patients, have shown significant improvement in visual acuity in the eyes that were treated with these cells. In one patient, we saw the patient go from blind 20/400 to, in 1 month, to 20/40, which is good enough to see, to drive a car. By contrast, the untreated eye, we only were allowed to treat one eye. Those eyes remained unchanged or continued to show decline in visual acuity. So far, we're very upbeat about these results. However, that's just the tip of the iceberg. This was an easy indication because, again, the eye is immune privileged, but also a small number of cells in a very defined area. But there are many other indications. So, for instance, corneal repair. There are 10 million people who have corneal blindness. It's the most transplanted organ of any organ in the body. And it's mainly due, or at least in a third of the cases, to the endothelial failure. So the solutions to these corneal diseases are whole cornea thickness transplants. So what you're seeing here is your usual just transplanting the entire cornea. But more popular and more recently now, what they're shifting to is something known as DSAC, which means you just transplant the corneal endothelium on Descemet's membrane. And what we have now figured out how to do is make whole sheets of these corneal endothelium from embryonic stem cells. We can now freeze them and ship them around the world. And we've done all the characterization showing that they have the various markers you would expect. We've done global gene analysis and showed that they look almost and act are almost identical to normal corneal endothelium. So we're in the process of now studying these in rabbits to see whether or not we can actually imitate the DSAC that we would want to carry out in humans. We're studying a number of other cell types in the eye. So this one cell type called the retinal neural progenitor, which I'll talk briefly about. We're also making progenitors so that we can make new photoreceptors, so that we can actually connect, hopefully, up photoreceptors after you've lost them and actually reverse blindness, which only a few years ago everyone thought was impossible. And then also we're making ganglion cells, which are very important because that's very critical for people who may have glaucoma. So these retinal neural progenitors, it sounds like a big word, but it turns out that this is really important because we can learn a lot in the laboratory and in animals from these cells, things that we can't figure out on our own. So for instance, if we find cells that can do certain tricks and treat certain diseases, we can go back in and study what are these cells doing? What are they making that's helping? So in this case, what we looked at is, again, animals that had retinal degeneration. And you can actually measure the electrical activity in the eye just like you do for your heart. So you can do an EKG to measure the heart activity. But in the eye, there's something called an ERG. And what you do here, the A wave is measuring the cones and the rods. Those, again, are what we see with. And we found that by injecting these retinal neural progenitors into animals that were going to develop this blindness, we could actually see here that from the untreated animals that the wave is very bad. But in the treated animals, we got this significant reversal of the progression of the photoreceptor loss. And if you looked at the histology, what was going on, what you'd see is the untreated animals, this little teeny black line here, that's what's left of the photoreceptors in these animals. Again, they're blind, very few cones and rods. The animals that we injected with these cells, here you can see, this is the outer nuclear layer, very robust layer, the cones and the rods are still there. So we thought, oh, okay, they're going into the eye and fixing it, and we were wrong. That's not what was going on. So you can do some DNA tests where you can actually measure whether the cells in any part of the body, or in this case the eye, were mouse cells or human cells. And it's called PCR analysis. So we can show here that when you take the mouse eye, all the cells in the eye are mouse, and there are no human cells in that eye. And this is very, very sensitive. It can detect just a few cells. So it meant that none of the cells will go into the eye. But when we inject these cells into the animals, into the tail vein, for instance, it turns out they get lodged in the lungs, they get filtered out of the blood, and they go somewhere else. And so they were protecting the eye from a distance. They were exerting an endocrine effect from somewhere else in the body that was rescuing the cells in the eye. And then we thought, okay, well, we know that this population of cells earlier on doesn't have that potential, and then a few weeks later it does. Well, what's changing in those cells? And if we could find that out, we can then just put a cocktail together with those molecules or cytokines in there to do this without the cells and to do it probably possibly even more efficiently. So if you look, there are all sorts of studies you can do with these antibody arrays, and so you can measure what's going on. So you can see this is the before and this is the after, and you can see here one group, there's a difference. You can see here's a difference here. So you can see the differences, and based on that, we now have a tool that can help us use this, not only perhaps just for this kind of degeneration, but any kind of neurodegeneration, whether you have any of these neurological diseases that you're losing neurons, and there's literally dozens and dozens of those types, so that once we understand what's going on here, we may know how to put together that cocktail to treat a range of diseases. But in the meantime, we're trying to figure out how to use the cells to do all sorts of neat little tricks. So one of the things is that we want to use these stem cells sort of like nanoparticles that can go in and fix the body the way we want. And it turns out when we were embryos, there was this cell, sort of like a unicorn, that had the ability to give rise to our whole vasculature and to our immune system. And these are known as hemangioblasts. And here they are, they start out from the master cell, and it turns out that as adults, we really don't have those in our body anymore. There are some cells that sort of resemble them, but they aren't the real trick. So what we're trying to do here is a new paradigm in biology, and that is to get a cell type that we don't have in our body as adults, and take a cell population that exists transiently in an embryo, but in the Petri dish catch that cell and expand it and use that to repair adults. And that's exactly what we did here. We found a way, a trick, to make these hemangioblasts and make very large numbers of these, and then test them in animals to see what they could do. And they could do some pretty amazing things. So in one case, we wanted to test these in a mouse model of what's called ischemia reperfusion injury, where you can actually put pressure on your eye, and that pressure in the animals will actually damage the retinal vasculature. And so we sent some of these cells that we labeled with GFP with a green dye so we could track them, and sent them down to Maria Grant down in Florida, and she injected them into these ischemic reperfusion injured animals. And what we found is that within 24 to 48 hours, here you're seeing these green cells going into the damaged retinal vasculature and fixing it. So that was pretty amazing that they could find a way to the eye. What was really interesting is in the uninjured eye, or in control eyes, there were none of these cells. So these were really smart cells, and they knew exactly where to go. And so again, these cells are able to go and sniff out areas. Say you have collagen-induced arthritis in a mouse, they can find that area. Here's another indication of what they can do. This is a mouse that has ischemic limbs, which means they don't have blood flow to the legs. So many people who have diabetes, will get limbs amputated, and the peripheral vascular disease, you lose your toes and everything. And so the goal here would be is can we use these cells to rescue the vasculature and rescue those cells? So in these mice here, this is the hind portion of the animals. This is the tail, and these are the legs. And this is a Doppler flow, which measures blood flow. And what you're seeing is there's no blood for that going to that leg. And that's a leg that would probably have to be amputated. One injection of our hemangioblasts within a month, complete restoration to the blood flow there. So someday, in the not too distant future, people are going to look back at our age and go, can you believe those primitive people? They used to cut people's legs off and just give an injection to the cells and they'll restore the blood flow and they'll walk away. And this is what we're talking about. We're talking about a change where this technology could potentially revolutionize medicine. Here's another illustration of the power of these cells. This is a myocardial infarct model. This was work that was carried out at the Sloan Kettering Institute with Malcolm Moore's group. And these animals that had heart attacks, we were able to, with one injection of these cells, to cut the death rate after a heart attack in half. This is a spontaneously diabetic rat. Again, back to our retinal vasculature. And here, we label these hemangioblasts, and you see them repairing the damaged, retinal vasculature. Diabetic retinopathy, as you know, is a severe problem with diabetics. But not only that, these animals, almost certainly, as you know, diabetes has a range of horrific secondary complications, and they're all microvascular. So not only are these cells probably fixing the damaged retinal vasculature, but if there was kidney damage or heart damage, those cells are almost certainly homing to those sites of injury and trying to repair those as well. So there are a lot of potential applications for these cells. So, as I mentioned to you, these cells, can repair vasculature. So here, again, if you're in the Petri dish, you can see they make these little capillary structures, and they uptake LDL and the different things that vascular cells are supposed to do. But as I mentioned earlier, these are bipotential. So the exact same cells, if you change the culture conditions in the Petri dish, you can change them and to make hematopoietic cells. So we can not, instead of becoming vascular cells, they become blood cells or immune cells. And so we figured out how to make entire tubes of red blood cells from these. So again, these red blood cells actually transport oxygen just like normal transfusible blood. We can get them to enucleate just like normal red blood cells. We've been able to make all kinds of blood types. We can make A blood, B blood, O blood, Rh, positive and negative. As a matter of fact, we have now embryonic stem cells and iPS cells that are O negative, which is the universal blood type. So when we make red blood cells from those cells will match everyone in this auditorium. So again, we now can make universal cells using this approach. Another program we have is to turn these cells into platelets. So the way platelets happen in your body is that you start out with these progenitor cells, and then they become these giant multinucleated megakaryocytes. And those megakaryocytes start breaking up into these little proplatelets, and then they become platelets. And so we now have been able to make large numbers of these platelets, very pure. And so, we've met with the FDA on this, and we've actually been able to show that these cells, whether you start with embryonic stem cells or iPS cells, they form clots and retract. They incorporate into developing clots or thrombi in animals, and we've been able to show that they circulate in animals nicely. So you can actually examine the platelets that we make using this technology and show that they're almost identical, virtually identical to regular platelets. So this is a cross-section of a platelet. This is an electron micrograph. It turns out that the platelets are biconcave, so this is two different cross-sections. And these are the platelets that we've made from iPS cells, and they're virtually identical. They have all the structures you would expect, the dense tubular structure, and they have the glycogen granules and all the organelles like mitochondria. So again, these normal platelets are discoid like this, and you can see the little hollow circles. And so we can show here that they're biconcave, just like they're supposed to be. That we can show here that they're the right size. So they're identical in all those morphological ways. They do all the things they're supposed to do. So if you have a cut, for instance, the platelets, they have to stick. So here we can show they stick and they expand. So these cells appear to be doing all the things we need platelets to do. And the problem with platelets, as you know, is they only live a few days. So there's always a shortage, and and in case of an emergency, BOTR, one of the Homeland Security, has a keen interest in what if a nuclear bomb goes off or a radiological weapon, and a million people or millions of people are affected. Where would we ever get the platelets? Blood lasts longer; it lives a few months, but platelets only last a few days. What we can do with this is we can freeze down those megakaryocytes I had showed you, progenitors, and make as many of these as we want. We could make enough for 2, 3, 5 million people, have them frozen down, and after an accident, within a couple days, have fresh platelets for everybody. So that's why they're so keenly interested. So we're currently working right now on these different tricks. In the body, you have blood flowing, so there's different forces that help this process occur. So this is something we can imitate in the lab using something known as microfluidics. But we're working on other techniques, certain bioreactor systems that allow us to make large amounts of these so that we can treat large numbers of patients. So again, this is all in process, and so hopefully someday we don't have to go to the Red Cross to give you blood to get platelets and red blood cells. And this is another cell type that is really exciting, probably one of the most studied cells that there are in the stem cell field, something known as the mesenchymal stem cell. And these are cells that normally live in our bone marrow. Also, you can get them from your fat, so you probably heard all these things about, you can use your fat to get these cells to rejuvenate this or that, and there's a lot of hype around this technology, and overseas people are going to use to get these cells. And it turns out that these cells normally can turn into fat and bone and cartilage. But what's exciting for using these in patients is that they are special cells. They're immune privileged, which means I can take a cell from me and put it in you and not worry about rejection because they can influence the immune system. They can modulate the immune response. Also, as with some of our hemangioblasts that I mentioned earlier, these cells can migrate to the site of injury. So you can put them into the peritoneum, into your belly, or inject them IV, and they'll go to the site they're supposed to go to, and they can facilitate tissue repair. So this is a little outdated here, but there are about 300 clinical trials in the US now using these kind of cells for a whole range of different indications. The thing is that they're all using adult bone marrow derived, and you have to go into your bone to get the cells. They can only be expanded at a very small amount, so there are really very significant problems with that, and in many of the clinics overseas that are promising stem cell therapies, they're saying they're going to get them from your fat, but I mean, very small number of those, if any of those, are really MSCs, and even if they are MSCs, they've been expanded to the point where they probably have no biological efficacy, so this is why there's a lot of caution about going overseas for these new unproven therapies. So what we can do with our embryonic stem cells, though, is that we can make as many of these cells as we want and have them ready off the shelf for when you need them. So we can make virtually an unlimited number of these cells. They're very easy to make. We can expand them to huge numbers. So I always like to use this Fenway Park analogy. If you think of how many MSCs you can make from one unit of bone marrow, that could be Wally, one Red Sox fan. From our embryonic stem cells, we can fill all Fenway Park. We can get 30,000 times more cells from one unit of our cells than you can from bone marrow. And not only that, but they have greater potency. So when you expand ones from bone marrow, often after you expand them, they're great negative controls because they don't work. So we have these cells that we can make. So this is great. Theory, you get a ton of these cells, but what can they do? So one of the really nasty diseases that affects people is multiple sclerosis. And there's a mouse a model called the EAE model that imitates this multiple sclerosis symptoms. So in these untreated animals, they have a clinical score of 2 to 4. That basically means that they're dragging their limbs, they're either completely or partially paralyzed. And what we can do is one injection of our MSCs, and these animals have a clinical score under 1. They're no longer paralyzed. They're jumping around in the cage almost normally. So we've been able to show that these cells work not only to prevent the onset of MS, but even after they already have it, to reverse it. And we've studied these extensively in the Petri dish. We show that they inhibit the immune system, the T cell function. So, with all these autoimmune diseases, your immune system that's hyperactive, we're showing that this can inhibit that. We've also looked at what these cells are making, and we see that they make different types of cytokines or different types of factors that can be very important for treating diseases. Another thing is that I mentioned to you these cells can home to the sites where they need to fix the tissue. Well, it turns out the adult bone marrow ones don't do that. So when we tried the adult bone marrow in these animals, we have very minimal effect, if any effect at all. So that our cells are working in this particular model far better than anyone would anticipate, and certainly better than what's available from adult tissue. And finally, we find that these cells have very unique properties that, like if you do the bone marrow, they'll go to the site of the injury, but they won't leave the vessels. They'll just sit there. And we find that our cells actually can what's called extravasate or transmigrate and get to the tissue to affect repair. So this is really exciting, and this is clearly something that we want to pursue and try to get into the clinic with. Another model that we've studied these cells in is lupus. This is systemic lupus erythematous, or in short, just lupus, is an autoimmune disease that affects virtually every organ or system in the body. So there's no cure for lupus. And it most often affects hearts, the skin, the lungs, blood vessels, liver, kidney, again, all the parts of the body. And in half of those patients, they develop something known as lupus nephritis, which it affects the kidney. So if you look at a normal kidney and the glomerulus, this is what you would see. But in a patient with lupus, here's the immune complex. What you're getting is the autoimmune process that deposits these into the kidney glomerula that leads to kidney dysfunction and failure, and without a transplant, those patients would die. So there are these mice, these NZB mice, that spontaneously develop lupus. So this is really the gold standard for testing lupus therapies. And when we went into these animals with our MSCs, the animals that were not treated started dying pretty dramatically, and we saw that one injection or 2 injections of these cells dramatically impacted those cells, those animals, so that in the first few months we didn't see any of those animals die at all. So again, these cells are really have the potential to do some pretty amazing things. That's just 2 diseases I'm giving you an example of, but again, there are literally hundreds of diseases that these could be used for. Again, I mentioned over 100 autoimmune diseases alone, not to mention multiple sclerosis, lupus, osteoarthritis. We're studying that now in dogs as well. Crohn's disease, irritable bowel syndrome, all the different organ systems, be it the liver, the kidney, and pulmonary. So there's really enormous potential for these cells. So to realize the real potential of embryonic stem cells, you really need to learn how to reconstitute those cells into more complex tissues and structures. And that's the whole field known as tissue engineering. And so we've now started to study how to reconstitute these cells to grow whole organs. So this was a study here that I carried out with Tony Atala's group, who he actually just cloned that bladder that I just actually showed you here. He actually grew that up in the laboratory and first studied it in dogs, and he's now using it in patients. We were in patients who have had these organs that have been grown up in the lab. But we were trying to do this with the kidney here, and these were actually, again, skin cells we took from cows. We wanted to test it in the cow. We grew up these renal progenitors, and we seeded them onto the synthetic scaffold, these little tissue-engineered kidneys, and placed them under the skin to imitate a kidney. And indeed, after a few months, what you could see in the cloned cells here, you see the straw yellow material. That's indeed urine. It meant these cells were taking toxic materials out of the blood of those cows and removing it. And so we saw elevated amounts of creatinine and urea. So again, these were really working. And then when you do the histology, you could see the cells that actually assembled into the glomeruli and into the tubules, and they actually connected up to the membrane in a unidirectional fashion to concentrate those toxic materials. And then finally, of course, we all know this controversy associated with embryonic stem cells. And the reason is that the usual way it works is you take an embryo that's tiny before it actually has any body cells in it, but you have to, these are leftover embryos in IVF clinics, and you have to destroy the embryo in order to make the embryonic stem cells. So that's what causes lots of grief in the pro-life community. And, you know, and there are some religious objections. So what we did is we found a way to actually make embryonic stem cells now where we don't need to destroy or harm the embryo in any way at all. So there's routinely in IVF clinics a method called PGD where you just take one cell out for genetic testing, and we found that we could just take that one cell out, and actually here you see an embryo at the 8-cell stage. We can pluck one cell out, make our embryonic stem cells, and the biopsied embryo continues to grow and become a normal, healthy blastocyst that can be frozen down or implanted to give rise to a child. And there have been millions of children born using that method. So we know that it works, and more importantly, we can make these embryonic stem cells that have all the markers and that turn into all the proteins. So we can now make these cells without harming any embryos. And then finally, I just want to thank the people who did all the work. I just get the credit for it. Thank you.
Thank you, Dr. Lanza, and we do have some time for questions, about 10-15 minutes, so we'll have some questions.
Okay. So, anyone have any questions? Yes. So the question was how long would it be before these are actually put into effect in human beings? So the answer to that is that it depends on the particular disease. As you know, as I mentioned, we're already starting in the eye for macular degeneration, but also we're hope that we're going to get into the clinic for, say, lupus or multiple sclerosis in the next year or two. And then, of course, depending on the disease, it may take more time. So it's going to differ from disease to disease. Parkinson's disease. So she asked, what about Parkinson's disease? We know, that's an excellent question. We can actually now make very large amounts of dopaminergic neurons, and in fact, these have been actually tested and made from embryonic stem cells, and they actually work in rodent models. So I believe there are several groups, and we're actually working with some of them, that are hoping to get into the clinic for that as well. Yes? Yeah. So the question was, how does, I guess, this playing out with the bioethics community? And, you know, I had actually started out in medical school, you know, trying to cure diseases, and never thought I'd be having to deal with all these theological questions. But yes, at first, of course, you know, I wasn't in a position to really be taking side with one religion or the other. But some of the religions obviously are opposed to destroying the embryo. So I think we've solved the problem of destroying the embryo. There's still some objections to some groups to even IVF, to even using that approach. The new iPS cell technology, of course, bypasses almost all of those ethical objections. Yes? Dementia. Actually, yeah, you're absolutely right. So she was asking, about, we have this application and that, what about dementia, right? Alzheimer's disease. As a matter of fact, that's an exciting possibility. We're actually starting that right now with one of the top groups in the world. We do think there's very good reason to believe that these cells should be able to impact that. So we're starting that work right now. Yes. Okay. Yes. Yeah. Yeah. Okay. So the question was, as she was mentioning the gaur that we had cloned, and then she was saying that an animal's dead, it's meant to be dead. There's some ethical questions here, and of course there are a lot of ethical questions. In that particular case, this was an endangered animal, and this was an animal that had just died, and so what we were doing was simply bringing back genes that